What is life? What is the difference between something that is alive and something that is not alive? Or something that was alive and now is not, so is dead? This is actually one of the key questions in biology. And it's something that I want to explore because very often I find myself explaining life to students, and actually it's quite embarrassing because we usually present a list of characteristics. For example, if you look at a textbook, or you go on an online encyclopedia and you look at what life is, very often you get a list of characteristics. This is what life does. Living things are characterized by the capacity for, and we see this list. I'm not saying it's wrong. I'm just saying it's a bit superficial, okay? So living things do self-regulate, this process that we call homeostasis, so keeping things the same. They're organized, they have structure. Metabolism is the sum of all of the biochemical reactions, all of the chemistry that's happening within a living thing to keep it alive. Living things will grow, they get bigger, they get heavier, they get larger, they occupy more space. They adapt. They respond to their environment, they respond to stimuli. Living things reproduce. And very often it's said that living things have a sense of purpose, or they seem to be doing something, which is a concept that we call agency. So these are characteristics of living things, but some of these are also characteristics of things that we understand are not living. For example, the capacity to grow is a characteristic of crystals. It's also a characteristic of fire. Fire grows, it spreads, it responds to its environment. Fire will also reproduce. We can see the sparks coming off this fire, and it might reproduce by starting new fires. But do we really think that these are alive just because they show one or two characteristics of living things? I think that most people would agree that these are not alive. Now, I have a particular problem with the last one here, agency, the sense of purpose, because superficially it does seem to be an attractive idea, that living things have a sense of purpose, they're doing something. And in fact, this was the basis of a lot of the philosophy of what is life, particularly in the 1970s. But in the modern world, I think we have various artifacts, things that we've created that show a sense of purpose, but they kind of make us rethink this idea that because something evokes a sense of purpose, it acts with a sense of purpose, that means that it's alive. This robot vacuum cleaner, for example, does not show any of the other characteristics of life. It doesn't have the full set of characteristics. It can't reproduce, for example. So in the natural world, there are clearly situations in which organisms seem to have a sense of purpose. These bumblebees, for example, are pollinating this lavender, and they're doing it because there's nectar and pollen. And okay, but the point is that they are acting, they're moving, they're doing something, they have a sense of purpose, and they evoke that sense of purpose in a human observer. But I'm going to show you a video by an illusionist called Marco Tempest. He works with the Magic Lab in New York City. And as an illusionist, he actually makes a specific point that we can make machines that evoke this sense of agency in us as human observers. It's a type of anthropomorphism. It's a characteristic of us that we impose on an artifact. So he actually talks about these machines appearing to be alive, or the group of machines acting together, responding to his input, to his stimuli, and this creates an overall machine that appears to be alive. So when it comes to using agency as the basis of a definition of life, what life is, it's very problematic, especially seen in the context of modern technology. This, again, seems to be a fairly superficial characteristic of life and doesn't really get to the problem. So when it comes to using a sense of agency for the definition of life, there's a risk that we become like a cat that's chasing a piece of string or a laser pointer. There's movement, we respond to it, we think that it's doing something, and we treat it as if it's a living thing, but actually it doesn't get down to the problem of what's making it move, what's actually happening. The cat really doesn't realize about the laser pointer and the person that's pointing, And that's what I want to get to today. I want to understand the laser pointer and the person that's pointing it. I'm not so concerned with the superficial characteristics of life. So essentially, to sum up this first part, these lists of characteristics of life really just scratch the surface. They don't go down to the mechanism. They don't look at what's actually happening to produce those effects. And as I've written here, this is from a paper that I wrote that I published last year. And it basically starts off by saying that these are secondary phenomena. They're emergent properties, and they don't look at the actual mechanism. And that's what we want to look at today. So there are many definitions of life, what life is. And the first thing to say is that some of these are working definitions. They're definitions that help us to understand a specific situation. For example, NASA's definition of life is this: life is a self-sustaining chemical system capable of Darwinian evolution. What that means is that if we get a sample from another planet and we examine it, we can look for chemical reactions and see if they're self-sustaining, if they keep themselves going. Or we can look for evolution if things are changing and responding to stimuli. And that would be a good sign of living processes. There is a problem with this. There are problems with many definitions of life, but this is a pretty good definition, but there is a problem. It relies on Darwinian evolution, and Darwinian evolution is a very strongly supported framework in biology. Every time we investigate Darwinian evolution, we find more and more evidence that supports it. I'm not saying that that's the problem. The problem is that Darwinian evolution is a phenomenon that affects groups of individuals. It affects populations, it affects species. It's based on the idea that a range of individuals will have a range of different characteristics, and some of them will be suited to survive in particular environments, and some will not. And the ones that survive pass on their genes, their information, their way of life onto the next generation. So this is not about the individual. And it's very strongly dependent on looking at generations. This is fine if we can culture some kind of sample from another planet in a petri dish and see it evolving, see it changing generation after generation. But it's actually quite a problematic definition, because we might get the culture conditions wrong to see that generational evolution. The other thing is that it's not looking at the individual, and really the question that is interesting to us is what's happening inside a living thing, the single individual, to keep it alive. So in this particular case, if we use this definition, for example, let's say that aliens came down to Earth, they ignored everyone else, they just took me away, and they looked at me. If they use this definition, they wouldn't actually know if I'm alive or not, because they wouldn't see me reproducing generation after generation, and they wouldn't be able to see this effect of selection from a group, a pool of individuals. With this definition, they would look at me and they would say, well, could be alive, okay? Don't really know according to this definition. We need a way of understanding single organisms in the moment. What is the state of being alive? So the first point that I'm making here is that there's a difference between definitions and an approach to life which looks at long-term processes, multi-generational processes, which can be fine in specific circumstances, but that's very different from looking at immediate life processes in the moment. That's really what we're interested in today. Now, this is an illustration of these immediate processes, the immediate state of being alive. So I really want to put emphasis on this. We're interested in what's happening within an individual organism. It could be a single-celled organism, such as this amoeba, or it could be a multi-celled organism, such as myself. And we want to know what's happening to make it work. So the question essentially is there a single characteristic, one defining characteristic that lets us make two groups of objects or two groups of situations in which we can say definitely that is alive, that is not alive, okay? Because alive, not alive, that's a binary choice, and we need a single thing that distinguishes these two choices. Is that possible? Well, there are some indications that there are properties of living things which are pretty fundamental to how they work. The first one that I want to look at is, okay, I have to mention some physics terminology here. I'm going to talk about entropy, but entropy is basically the amount of disorder in a system. So when things are more ordered and more structured, such as a living body, it has less entropy. So this is an observation by Erwin Schrödinger, who is a famous physicist, famous for a range of different contributions to physics. But as a biologist, I think that his main contribution to our field is this observation about entropy and about order in systems. He said that the universe is characterized by increasing disorder, or entropy, increasing disorder with time. In fact, we can see time on the x-axis here. Over time, the universe tends to become increasingly disordered. Okay, so things tend to fall apart, things tend to become more disordered. The main characteristic of life for Erwin Schrödinger is that living systems, at least locally, within the context of the universe, locally, they decrease entropy, they increase order. So what that means is that we go from a state where things are sparse and widely spaced to increasing structure. And here, of course, we're talking about atoms and matter, the matter that we find in the world around us. So living things fundamentally aggregate and organize and create structure. So for Schrödinger, life is a process that orders matter, always in that context of a universe in which matter and energy tend to dissipate. So basically, biological organisms are using external matter, using external energy to accumulate organized structures. And we can tell that this is basically true. Think about a tree growing in the sunlight with rain falling on it. The wider system is a high entropy system because we have water falling, water dissipating everywhere, heat, light, everything dissipating. It's a high entropy system. But within that context, the tree is taking light, carbon dioxide from the atmosphere, water and nutrients from the soil, and it's getting all of that matter. So we're talking about atoms, and they're making structure. It is literally the embodiment of reduced entropy. So this is a key characteristic of life. It creates order and it reduces locally entropy. The trick is in how it does it. Okay, before we really start and we go into the definition of life, I also want to look at some work by Professor Adi Pross, because I think that many of the things that he has said in his book here are basically true, and they actually form the basis of what I'm going to go on and say. And I think that really, if I have anything to add, it's one detail into this, but it's based on these concepts that we'll see here. Now, first of all, I should say that Professor Pross has a long-term view. He's very interested in looking at the origins of life, rather than what I'm trying to do here, which is the immediate state of being alive. Now, the two things are linked, but it means that the point of view here is very definitely long-term, based on generations, and there's an element of evolution and natural selection, which we've already discussed. But there are some characteristics here which I think are, like Schrödinger, fundamental to life, and it's important that we understand them before we go on and look at the rest of what I have to say. So the first thing is the concept that life maintains stability by a dynamic kinetic means. It does it on its own, it's autonomous. What does dynamic kinetic mean? Well, I can illustrate this by giving you the opposite case, which is chemical inertness. Imagine a diamond crystal, so a block of carbon atoms. Now, those diamond crystals can be stable. They can be stable for billions of years, and they're stable because they're chemically inert. They don't tend to react with things. So that's one way of being stable, but they don't do anything. And this is a big difference. Life is stable. It creates a form, in my case, the form of my body, in the case of the tree, the form of the tree, and it does it actively, continuously. Bits are falling off, but it puts bits on, okay, if I can say it in that way. So it's about maintenance, repair, growing, and kind of an active approach to having a stable body. So the first point here is that life maintains stability autonomously on its own by dynamic kinetic means, by moving, by doing stuff, okay? So that's a key characteristic of life. Another characteristic is that in the past, and here we're really talking about the origins of life, it seems that there were molecules, chemical substances, which could replicate, they could copy themselves, and they could also perform functions. I talked about metabolism before, the biochemical reactions that keep things working. So it seems that in the past, there was a process whereby molecules gradually started to copy themselves and to do work, to do reactions that helped each other. They help each other. So it's not that in life there is one particular molecule, one particular structure that does everything. It's not that DNA does everything, for example. It's all a network. There are different chemicals which are basically helping each other. And life has come from this replication reaction. So the capacity to copy these molecules has basically kick-started the whole process of life. As the title of the book says, how chemistry becomes biology. We also have a term for that, which is abiogenesis. So the idea is that abiogenesis, the origin of life, is based on this replication and metabolism and molecules helping each other. The other thing is that molecules are variable in their structure, especially what we call macromolecules, big molecules. They can have some variation in the number and position of atoms, and they can still work, maybe a little bit more efficiently, a little bit less efficiently, but they are variable. And the idea is that even at the level of chemistry, molecules, natural selection will favor some forms and not others. So it seems that the complex, sophisticated systems that we see in the modern world have evolved from simpler systems, but systems that are still undergoing natural selection. So the concept that comes out from this is that over the geological timescale, molecules and systems, reaction networks, as we call them, have become increasingly more and more complex and sophisticated. And I think these are key characteristics of life. So the point of Professor Pross's book wasn't exactly a definition of life. It was more a consideration of the various characteristics and how they've come about. But he did provide this sentence, which is essentially a definition of life based on these concepts. He said that life is a self-sustaining, kinetically stable, dynamic reaction network derived from the replication reaction. Now, I'm aware that that's a bit of a mouthful. There are lots of concepts in there. And basically, I'm trying to summarize a whole book here, a whole book full of really cool ideas. And it's difficult to do that with one slide. So what I'm going to do is take that definition, put it at the top, and we'll go in reverse. We'll look at each part of that definition and break it down. In a sense, we just built it up, but now we're going to break it down, and that way, perhaps we'll have a better idea of what it actually means. Okay, so the first point is that life is derived from the replication reaction. The replication reaction is the tendency of some molecules to copy themselves. So life is derived from the replication reaction. Without replication, everything stops after the molecules wear out. They don't last forever. The other thing is that life is kinetically stable. We already talked about this as being the opposite, in a way, of chemical inertness. So again, the diamond that doesn't react, that's a kind of inertness which creates stability, but life is kinetically stable. It's working to create structure to be stable. Then life is a dynamic reaction network, So it's a network of molecules that reacts. Now, why have I put this in bold? Why have I put this in bold? Life is a dynamic reaction network. I'm not saying that it's not. The reason why I put it in bold is because I think that here there's a detail missing. And this is the solution. It's also the problem with understanding life. Because actually this reaction network, what is it? What is the reaction network? Now, it's not that we don't know what this reaction network is. The problem is actually that we know precisely what this reaction network is, and we know how complicated it is. Any student of biology will spend years studying the various components of this reaction network. They'll study DNA, they'll study RNA, they'll study carbohydrates, they'll study membranes. All of these words and terms represent structures and processes that we know in a great deal of detail. And the problem, essentially, is that we know there's a vast range of different structures that seem to work in very, very different ways. They have different, they're involved in different chemical reactions, they do different jobs within the cell, and we have this vast range of detail, and it's very difficult to pick out one single characteristic that defines them. Don't forget that we're looking for one thing that defines a state of being alive, a common principle governing all of these disparate different biological molecules and the way that they work, but one thing. Biological molecules are very, very different, certainly in terms of their chemistry. Chemistry is the interaction between different atoms, between different molecules, but they do share some characteristics at the level of the underlying physics. They are atoms, they have electrons, the components of the atoms move, they spin, they oscillate and vibrate. And basically, what I'm saying is that we have to look at a level a bit before chemistry, okay? In fact, very often we say that biology is based on chemistry and chemistry is based on physics. To understand biology, we actually have to go to the underlying physics. This is where it all becomes common ground. The really good thing about living in the modern world is that we can actually see these things directly. And what I mean is literally see atoms. We have the machines, okay, the microscopes, to look at atoms, to look at molecules, to look at the full range of scales from atoms to organisms. And in fact, later on, we'll be seeing the results from one particular study, which uses a high-speed atomic force microscope, which is basically a probe with a very, very sharp point. It goes down to one atom, which vibrates. And you can scan that over a molecule, and you can actually see it moving. This is new stuff. It's very, very cool. And so we can actually get a video of things moving and things working. And it's at that kind of level where we have to look. But we have the instrumentation to do that. We have the possibility to do that. So the first thing is to get over something else that you were taught at school, which was probably the Bohr model of the atom, which is a very good way of visualizing the atom, thinking in terms of charges, the orbits of electrons, that kind of thing. But this is an old model from 1912. It's not something that we use anymore. Nowadays, we appreciate atoms in terms of quantum mechanics, and it gets a bit difficult when we talk about quantum mechanics. Physicists tend to use mathematics to describe quantum mechanics, and it's very difficult to express in plain language. I'm going to attempt to do that. I'm not a physicist, but I will attempt to interpret the quantum mechanics of an atom in a way that shows the importance of these phenomena for life. So the first thing is I'm going to show you a different model of the atom, which we have to understand in order to understand life. And it's based on the fact that according to quantum mechanics, we don't have a precise position for each electron. We have a probability of finding an electron in a particular zone, an orbital, not an orbit, but an orbital. And what this means is that when we represent an atom, we see basically a cloud of probability where we might see that electron around the nucleus. So the next image I'm going to show you is a representation of one single atom, the most simple atom that there is, the atom of hydrogen. But you'll see that there are many different images because it represents different states of that atom, different energy states, if it has more or less energy. We'll come to what that actually means in a minute. But basically, a single atom can have electrons in essentially in different positions or different positions where we might find those electrons. Now, the point that I'm making here is actually that atoms are very dynamic. The electron moves. It's difficult to tell exactly where it is. It's difficult to detect it. All of the components of the atom are spinning or oscillating, vibrating. The subatomic particles within the protons and neutrons are also spinning. They're also moving. The whole thing is moving. And the first thing that we need to appreciate when it comes to the physics of life is that atoms do not stand still. Due to the uncertainty of where the electron is, due to the fact that it all moves, atoms actually vibrate. They vibrate. They spin. They have various movements that they can do, but they also oscillate and they vibrate in place. Just due to these simple physical principles that we can't really pin down where the various components actually are in any one moment, the practical effect is that the atoms are oscillating or vibrating. Okay, this is extremely important because when we have groups of atoms together, they jiggle. Now, I can use the word jiggle, which is a very silly word, but I've heard it used very often in this context. I can use that word because I'm in good company. If we look at the British Broadcasting Corporation's archive in 1983, they interviewed the physicist Richard Feynman, and he actually says jiggle. Okay, atoms jiggle. So this effect that we see means that molecules, groups of atoms, atoms that are linked together in some way, they're bonded, they form a coherent structure. These are moving all the time, they're jiggling, they jiggle everywhere. And this is a very basic energy that there is in all matter, in all atoms and electrons. So everything is jiggling. Even, for example, this desk, even though the desk itself isn't moving anywhere, it's not going anywhere, all of the component atoms within this desk will be jiggling in place. They'll be moving, oscillating, jiggling around. Okay, it doesn't mean that they actually do anything, but they are there jiggling. So molecules are constantly vibrating. They're constantly moving. We can see this agitation. And this is actually it's a movement, and this movement will increase if the temperature is higher. By definition, if molecules are moving around at a greater speed, the temperature is higher. Those molecules will be banging into our thermometer more rapidly, and they'll tend to transfer this kinetic energy to the thermometer, and the mercury in the thermometer expands as well, and we measure a higher temperature. So because it's basically about movement and motion, things being done, a dynamic situation, and about the level of agitation, the amount of agitation, which we measure as heat, it's about thermal motion. And so the field of research into this is called thermodynamics. Now, thermodynamics is a universal phenomenon. It applies to all matter. It applies to the matter in a rock. It applies to the matter that's inside me. So one of the effects of thermodynamics, this thermal motion, is that we can see things at small scales moving constantly. The individual particles will jiggle and move, but they're also bombarded by surrounding molecules. For example, molecules in the air or molecules in liquid. Water in particular, this is a good example. Those atoms can actually move relative to one another in different ways, which you can see here on the screen. And they're actually quite active. They're jiggling around all the time. Molecules of water are very small. There's many of them, and they tend to bombard things. And so we can see that a large, in this particular case, a large orange particle, we can see that basically bumbling around and jiggling around. In part, that's because of its own thermal motion, but it's also because of this effect of thermal bombardment. Now, this thermal motion and agitation is the basis of these little movements that we see at small scales. We call it Brownian motion after Robert Brown, the person that used for the first time a system of pollen grains in water and saw them jiggling around and understood. So nowadays we call this motion at small scales Brownian motion. And this is a kind of universal thing. Okay, thermodynamics is universal and Brownian motion happens everywhere. Now, Brownian motion is extremely important because it's actually the force that drives processes such as diffusion. Diffusion, the effect that particles will tend to move They'll move across a room in the air or in water. And in fact, in this slide, we can see over time, 1, 2, 3, a concentrated mass of particles on the left, the orange particles over time. They take this kind of random walk. We can see this red arrow, which is zigzagging everywhere, because Brownian motion is a kind of random motion. In fact, it was Albert Einstein in 1905, in one of his famous papers, that worked out exactly why this happens, because it's all a game of probability. If that molecule, if that particle is surrounded by water molecules, then it'll be hit from each side, and there'll be moments when it's hit slightly more times on one side with respect to another. So receiving more hits, more energy from one side, it tends to go in the other direction. And so we have this kind of movement, overall movement of the particle, following this random walk, and this is actually the force that drives diffusion. So when we get down to small scales, we see this random jiggling movement. Now, this is an example within cells. Well, now we're going to look at motion within cells in particular. If we look inside that cell, that's the motion that's interesting for us today. I'm not really too bothered by the fact that this amoeba is moving its extremities. I'm more interested in the tiny jiggly motions that we see inside. Now, if we look at any cell, we'll see this jiggling motion. Some of this apparent motion that we can see here is due to the microscope refocusing. But if we have a good look, and I'll try and use the laser pointer to point out some parts. For example, if we look by the laser pointer here, we'll see something jiggling around. All of these things are jiggling on a very, very small scale. So that kind of motion, that Brownian motion, is occurring within cells. Now, these are onion cells in which we can see a very definite movement. It's called cytoplasmic streaming, and it's a movement of the various components within the cell. Now, if you're a biologist, the first thing that springs to mind is that there's actually quite a well-known mechanism for these movements, which is based on something that we call the cytoskeleton. So a network of proteins that runs throughout the cell, which is involved in the transport of substances within the cell. But the point that I'm making is that even that is affected by the Brownian motion. Okay, so all of these components are subject to Brownian motion, to being bombarded by water, and actually that's really important in a positive way in helping these processes to occur. This is really part of the point that I'll be getting to. So here we see movement again within an onion cell, and we can see the cytoplasmic streaming, the movement within the cytoplasm within the cell. So within cells, one of the characteristics is motion. They're continuously in motion. Now, to give this a bit of context, what I want to do is to look specifically within the cells, and we're going to see structures such as this. They're plant cells where the what they're called microtubules of the cytoskeleton are visualized with this green fluorescent protein. So we can see that the cytoskeleton is a network that runs throughout these cells. So what I want to do is explore some of the actual components that work on the cytoskeleton, but also within the cells. The difficult thing about this lecture is that we have to understand biology in a very short amount of time. So we're going to look at a computer model which was made by the California Academy of Sciences, which will take us deep inside a leaf. This was published using a Creative Commons license, so I'm going to show you the whole thing, including the credits at the end. But basically, it's taking us within the leaf, and it's great because it gives us a context, a macroscopic context. And we're going to go into the tissues, which are groups of cells which are doing the same thing, in this case, the photosynthetic tissues of the leaf. And then we're going to go within the cells and look at the structures within the cells. There we're going to get down to the level of molecules. We'll see the cytoskeleton, for example, and we'll see the different molecular machines that are working within the cells. So we're going into the photosynthetic tissues here. This is the Palisade layer. These are the cells that are actually doing photosynthesis. In fact, we can see these bodies within them, which are the chloroplasts, which are actually the site of photosynthesis. So if we take a single palisade cell and we go inside it and have a look at these very bewildering structures, but we'll explain some of them in a minute. The first thing is there are strands running past us now. Those are the cytoskeleton. These are the microtubules and filaments of the cytoskeleton. And then we can see these bodies coming up. This one is a chloroplast, which is the site of photosynthesis. Now, photosynthesis occurs on a system of membranes called the thylakoids, which we're seeing now. And the thylakoids are covered in molecular machines, which is the bit that's really interesting to us now. So we're seeing these little blobs, which are actually what we call enzymes. Enzymes are proteins that do work within the cell. And we can see that they're spinning here. And what they're doing is they're actually creating a small chemical, which we can see as these wiggly lines, which is called ATP, adenosine triphosphate. We'll see this again in a minute. But the point here is that we're seeing molecular machines, and they're moving. They're in this environment which is wiggling and jiggling. We can see the ATP jiggling everywhere. They are exposed to this effect of Brownian motion, and it's actually a part of how everything is moving and everything is operating. So I'll just let this run until we see the end credits. This is a fantastic piece of work. Okay, so let's talk about these molecular machines. Now, the machine that we've just seen, here we can see a version which is actually upside down with respect to the video that we've just seen. This little molecular machine is actually called ATP synthase. It's producing a molecule called adenosine triphosphate, which if you read any biology textbook, adenosine triphosphate, or ATP, is what we call the energy carrier. We'll look at what that actually means in a minute. But essentially what's happening here is diffusion. We have a membrane. Everything is moving, of course. Everything is agitated thermally. And we have this membrane, and on one side, there's a high concentration of protons, of ions of hydrogen, which we can see up here. And a bit like a water wheel, a bit like the flow of water, those protons are moving, they're diffusing from an area of high concentration on one side of the membrane to an area of low concentration. And as they go through this system, they physically push it. There's a part of this ATP synthase, which is known as the rotor, because it rotates. It's just like a water wheel. Okay, it's a physical movement of matter that is pushing and turning a wheel. It's the analogy of a water wheel is very, very pertinent here. Now, what that wheel, what that rotor actually drives then is a system of proteins, which we can imagine like fingers, and they're moving backwards and forwards due to this system. They're taking the substrate, the basic molecules, which are ADP, adenosine diphosphate, 2 phosphates, and a little thing called an inorganic phosphate, and they're sticking them together. This movement actually brings the 2 substrates together in a position where they can bond with each other, and they're forced together, and it makes this bond, and it makes the adenosine triphosphate. To understand what's actually happening here and how adenosine triphosphate actually carries energy, we have to look at the molecule itself. And I say that the diphosphate and the phosphate group are forced together, and actually it's a bit like pushing on a spring, because the way that ATP carries energy is in a kind of kinetic way, in a sense. What we have are phosphate groups which are negatively charged. They have lots of negative charges. Essentially, this means that they have lots of electrons which are kind of free. They're free to make bonds, but they're also there together next to each other. And when you have, for example, 2 magnets, the same end of the magnet, and you try and put them together, they push against each other. Now, this will stay together as a molecule, as a structure, because the ATPase has forced it together, created a bond between the phosphorus atom and the oxygen, and that is holding everything together. But it's in a state of tension, like physical, kinetic tension. And if something comes along and breaks that bond, then that tension is released. It's like a spring, and it's actually a movement, a kinetic physical movement. And the way that ATP actually works, okay, again, if you look in a textbook, they'll say that it carries energy as if it's got a cup with some kind of sparkly blue liquid in it. But what actually happens is that these components will smack into other things like proteins. They'll bond with them, push them. There's a kinetic energy. They push them. They change the shape. They change the conformation of other structures. So in biology, ATP is very important because it can add itself to other structures and change their shape, and that activates them, that moves them, it gets them moving. Now, most structures within a cell work because they move. The molecules will change conformation, change shape, and so ATP is very important in getting things moving. So this is the way that it does that. This is the energy that it's carrying. This kind of process is key to a range of different phenomena within the cell. Here we have a very well-known phenomenon. If you're a biology student, you'll certainly study this. This is DNA transcription. It's basically reading the code of DNA. And what's happening here is that a protein body, which is doing work, so it's an enzyme, it's called RNA polymerase. What it's doing is opening up that molecule of DNA, and it's basically creating a strand of what we call messenger RNA. And that has a copy of the code that's encoded in the DNA molecule. Okay, we could spend hours going into the details of this mechanism. That's not the point for today. The point is that this is a system that works through motion, through movement. In this case, a linear motion. And the molecule that is organizing this, the RNA polymerase, is basically ratcheting along that DNA molecule. It does so with the help of ATP, which is smacking in there and changing its conformation. It gives it the energy in that way that we just discussed. Unfortunately, I don't have the rights to show you today a really highly detailed animation of this, but I suggest that you go to the website of Drew Berry, where there are a lot of detailed animations of how RNA polymerase actually works to do this. But let's just say that it's using the energy from ATP to create a ratcheting mechanism which sends it along the DNA in a linear motion and does the work. Now, this concept of ratcheting along, physical conformation changes that guide a linear process or a directional process, is something that we see everywhere in biology. This, for example, is the next step in that process. We take the strand of messenger RNA, and that is basically read by a body called a ribosome. Now, as we can see in the animation and in these papers here, the ribosome is in two what we call subunits, and they swivel with respect to one another, and they change conformation in a way that ratchets them along the mRNA molecule, and as it goes, it creates this protein. This is driven not by ATP, but guanosine triphosphate. It's another nucleotide. It still has a triphosphate. It still has the 3 phosphates, so it works in the same way. But ATP actually isn't involved in all life processes. Okay, that's not a universal thing that can explain everything. But this change in conformation and directionality in work, that is a universal phenomenon in all of this work, all of these reactions that we see within the cell. Now, we talked about the cytoskeleton, and it's not just a structural support for the cell. It's also a system of transport. It's involved in transporting bodies, particles, components around within the cell, helping the cell to organize itself. It's involved also in cell division because it helps the components of the cell to be reorganized to divide the cell. It's involved in a whole range of processes. And this is basically driven by what we call motor proteins. I'm going to present a couple of motor proteins now. These are motor proteins from eukaryotic organisms, so plants, animals, protists, so one branch of the tree of life where we have a lot of information about how this works. We're starting to appreciate basically that any cell that divides needs some kind of equivalent system in order to do this. So it seems that even, well, throughout the whole tree of life, there is some kind of cytoskeleton-like system for organizing the cell. But what I'm going to show you is from eukaryotes, from our branch of the tree of life, where we have a particular depth of knowledge of how it works. So what we're looking at here is one of these motor proteins called kinesin. And kinesin here, we can see it kind of walking along a microtubule, one of these trackways within the cell. And as we can see in this animation, it's doing so thanks to the involvement of adenosine triphosphate, ATP. And it's changing conformation and basically walking along this microtubule. Okay, so we'll come back to this in a minute. We'll look at some other motor proteins as well. This is the video that was created with the high-speed atomic force microscope. This microscope with a very, very fine point that vibrates and lets you scan across a molecule in real time as it's walking. So this is not a computer model. This is not an artist's impression. It's actual real data from the original peer-reviewed paper. So that data is incorporated into a model that we can see on the left, which is showing us how the myosin molecule is actually working, how it's walking along an actin filament within a cell. Now, something that comes out of this, a concept which I think is fundamental to understanding how this works, is that the molecule is moving along one plane. It's bending, changing its conformation along one plane. And that means that the overall direction in the system, in the process, is unidirectional. In this case, it's linear. There are other systems that are rotational, but here we have a molecule which is changing conformation in a uniplanar way, in one plane, and it's creating a unidirectional process. Another thing I'd like to point out here is the use of the word or the phrase rigid hinge. In this particular study, they talked about one particular part of the myosin molecule, this part here, which acts like a hinge. It's involved in what they call the power stroke when the ATP arrives and it changes the conformation and it produces this stroke which gets everything moving. So they talk about a hinge. This is something that I've heard very often in the literature in reference to these molecules. Hinge, something that bends, a conformation that changes. In fact, this is dynein. This is another molecular machine. And in fact, when Stan Burgess, who's one of the researchers that looked at the structure and the functioning of this molecule for the first time, when he talks about this, which you can see in this YouTube video, I put the link up there. When he talks about it, he talks about a hinge motion near what we call the microtubule binding domain, the area, the region of this molecule that binds with a microtubule. And he said that the hinge is basically here, a bit like an ankle on a foot. The way that dynein actually moves is very, very complicated. I've put in another link to another video by Ron Vale, who's one of the people that really started to look at these molecular machines for the first time. And that he's got a very detailed exposition of how dynein actually works. But basically, there's one thing that I'd like to draw attention to here. When we discuss how they move, the forces that make them move, ATP or nucleotides are involved. They change the conformation, and that powers them. It actually gives them the force to move. But something that's actually acknowledged in all of this is the existence of Brownian motion. So this jiggling, which affects everything at small scales. Now, one thing that Ron Vale said was that this could actually be a positive process because these molecules are basically dancing around, and this movement helps them to find a place to bind. It helps them to find a correct place where they can actually put down their feet and start walking. So some people see Brownian motion as a force to be overcome. Some people acknowledge that it could actually play a role in this movement. But there is actually another school of thought in the scientific literature where this Brownian motion is perhaps the main force driving things along. ATP is involved, it changes the conformation of the molecule, but the thing that actually pushes and gets things moving is this Brownian motion. So I want to discuss the involvement of thermodynamics in the movement of molecular machines. This is not my original thought. It's basically started off with a range of scientific literature, especially by Astumian and co-workers who investigated this idea that thermal activation, thermal motion can generate force within cells through the system. So what I've done here is I've taken the kinesin and I've added some water molecules, okay? Just red dots which represent the water molecules because they're always present. They're a context in which everything happens and it has thermal energy. And so everything here is jiggling. The motor protein is jiggling, the microtubule is jiggling, and it's immersed within this substrate, within this context of water, where all the molecules are jiggling. They're bombarding the motor protein, so it's all in motion. Now, if we look at the thermodynamics, there is actually a precedent for understanding what's happening here. What we see here is a system which is being bombarded. Again, we see the red dots, but the configuration, the conformation of this system means that when it's agitated, it tends to go in one direction. It's a little bit more complicated than this, as Richard Feynman stated in his lectures, which you can find here. They're published by Cornell University and the California Institute of Technology. You can go and check out the original lectures of Richard Feynman. But basically, the conformation of this system is key to getting it working. What I mean is, everything is being bombarded, everything is jiggling, and it can't actually be used to do any work because even the mechanism itself is jiggling. Here we have a ratchet, which is made of a gear, a toothed gear, and a pawl. So the whole thing is a ratchet with a toothed gear and a pawl. Now, Richard Feynman made the point that the pawl is also jiggling. And so if everything is at the same temperature, everything is jiggling, and the pawl can slip, the toothed gear can slip backwards, and no work can be done. Work in this particular case is shown by M for mass, which is a mass that's being wound up and lifted against gravity. So if the temperature is even across this system, then it can't actually really do any work. But Feynman made the point that if the temperature on the left at T1, if the temperature is higher, if the particles are moving around, bouncing around faster and hitting that structure, then it will actually be vibrating and moving at a faster rate than the pawl. So there's actually a good probability that when it's hit, the pawl will be down and the whole thing will turn in one direction. So essentially what this is saying is that there are circumstances where you have a structure, a physical structure, that can extract energy from this thermal environment. Now, as a practical demonstration of that, I want to show you a video which Jamie Wecker very kindly gave me permission to show. This is a video that you can find on YouTube, but she said that I could show it in this lesson. And what we're doing here is we're inducing a kind of artificial Brownian motion. We're giving energy to those, let's say, water molecules. But what they're doing is they're hitting the vanes on this blue wheel. And that's connected then to the gear, the toothed gear, which in this case comes from a clock. And there's a spring-loaded pawl stopping the toothed gear from going backwards. Now you can see that this chaotic thermal movement, let's imagine that it's thermal movement anyway, this chaotic movement is converted into directional movement. This is a key point. It's a machine that converts chaotic movement into directional movement. In this particular case, into a rotational movement. But the point that I'm making is that the machines that are working inside cells could be doing the same thing. They're being bombarded, they're jiggling, and their conformation, their shape, and the way that they're set up will direct this random thermal movement into a directionality, and this can be used to do work. So if we look at the motor proteins or the other molecular machines in this context, in this thermodynamic context, we can see that the thermal agitation could actually be a physical force that helps the whole process to have a directionality, to go in one direction. So we certainly have the involvement of ATP. Okay, it comes along, it changes the conformation of the molecule. So it comes along and hits that molecule and changes it. But then we have a kind of continuous movement. We have parts that swing around, parts that, it does happen very fast, but there is a continuous process always happening in the background, which is this bombardment, a continuous bombardment by the water. And in a sense, you can see the ATP as creating a new conformation, which is then free to be pushed by the thermal bombardment. So essentially what I'm saying is that the ATP, and don't forget that it switches between ADP and ATP, it's essentially like the pole, like the two different switching states of the pole. And it's something that essentially completes the configuration of the molecule, frees it up, lets it be, lets it move in response to the thermal agitation, and it's then the thermal agitation that is the main force pushing everything along. So a way of thinking of this is that the mechanism that moves, the thing that is actually doing the work, is like the lock. Okay, it needs to change its shape, it needs to change its conformation in order to work, in order to unlock the door. ATP is the key, or to be more precise, the switch between ATP and ADP are the two states of the key. Okay, left, right, on, off. And it's true that when the key enters the lock, it changes the conformation of the lock. All the little pins will move and they'll get into the right shape for it to actually work. So ATP really is like a key, but there is another force involved, something that's missing from this model of the lock and key, and that is the hand that turns the key. And that hand is thermodynamics. It's thermal agitation. It's the thing that gets stuff moving. It's the actual physical kinetic force that does things. So I'm not saying that ATP doesn't do anything. I'm saying that it's key. It literally is key. It's key to changing conformation shapes, to getting the movement started. It will give its own thermal energy to that and help to create a gradient, a thermodynamic gradient across the molecule. So it really gets things started. But then that's acted upon by the thermal energy in the environment. There are a number of points that I want to make that are linked with this. Firstly, ATP is not a universal thing. It's not found everywhere. It's not the key to life in all situations. So not all of these molecular motions within cells are actually powered by ATP or even by other nucleotides. It can't explain all of the motion that we see in cells. But thermodynamics is everywhere. That is universal. That can explain all of the movements. Even the movements of the membranes of the cells, that's a thermodynamic effect. That really doesn't have much to do with ATP. It's thermodynamics. So that really is universal. And even in the case of ATP, the original source of energy to create the ATP, as we saw, is basically a thermodynamic gradient. It's this gradient in concentration of protons, which is then essentially donated to the ATP molecule. Essentially, what we're saying here is that thermal agitation is a universal phenomenon that affects all atoms, it affects all molecules, and it can explain these motions that we see within cells. It can explain the working of the cytoskeleton. It can explain the movement of the membranes. It can explain the movement of the phospholipids that make up the membranes. It's actually involved in a whole range of phenomena beyond what ATP is involved in. So you might have noticed that this ratcheting mechanism depends on not being able to go backwards. So in order to take this random chaotic movement the molecules have to be configured in a way that they can't run backwards. And that's actually not true of these molecules. For example, the ATP synthase that we saw can run backwards. And there are different, let's say, forms, similar forms of enzymes which run in the other direction to use ATP. In one group of organisms, the archaea, there are even single molecules which are called ATPase synthase because they can do one or the other depending on how much ATP is in the environment. So these machines can run backwards. Even in the case of molecular motors, they can take backward steps. In the case of dynein, this is actually quite common. But one thing that is common to all of these different systems is that their conformations, their shapes change continuously due to this thermal agitation. And one way that I've heard this expressed in the literature, I've heard this a number of times, is that they sample the environment. They sample the environment with motions that have a topologically preferred directionality. So everything is wiggling around. And due to the interactions of charge, for example, with other molecules, there'll be one, and with themselves as well, there'll be one direction that is more probable, it's more likely. So they can run backwards, but they tend to go in one direction, either rotational or linear. The point is that we have a directional process. So despite being capable of backwards motion, all of these molecular engines are essentially exploiting thermodynamic gradients, either a gradient across a membrane or across the structure of the molecule itself. And this drives a uniplanar conformation state change. So uniplanar means in one plane. For example, they're rotating in one plane, or they're moving backwards and forwards, they're hinging in one plane. And this creates a rotational overall process or a directional processive overall process. And that directionality can perform work. So if there's one single phenomenon that characterizes living things and delimits them from non-living things, it's the way that their molecules are configured to take random thermal motion and direct it. And when you can direct it, you can create work, you can do things. That's the one single difference between living systems and non-living systems, the conversion of random to directional motion, which can do work. So here we have a diagram, which on the left here, we see over time a system which can dissipate energy because all of the various molecules, the water, which is shown in red and white, and I've thrown in there an organic molecule. It's a very simplified cartoon, but what we can see over time is that this thermal energy actually doesn't do anything. Everything is moving. The water molecules are colliding with everything. The organic molecules are bouncing around, but nothing actually gets done. We can contrast that with a living system. Now, I've based this very loosely on a motor protein, but it's a cartoon. It's not to scale. Over time, what happens is this thermal agitation is actually directed into movement of the molecule, the motor protein, but also a processive directional movement in the process. So here we have a net difference between an energy dissipating system and an energy directing system, where the thermal agitation and the ATP and the nucleotides, the chemical energy, it's all used to induce a directional motion. And that is the key. So this represents one difference, one single difference between objects that are alive and objects that are not alive. So objects that are not alive are basically not doing anything with that thermal motion. The thermal motion is random, there's no overall directionality, and there's no work that's done. Whereas objects that are alive use that thermal motion either in a way that makes a rotational movement or a linear directional movement, and that movement can be used to do work. So this is the one difference between living systems and non-living systems. So firstly, with regards to the thermodynamics of this situation, I'd just like to repeat, because sometimes this gets criticized because there's this concept of negative entropy, which really isn't actually a problem because the whole thing is existing within a positive entropy system. And I think the best analogy of that is a water wheel. It's lifting a small quantity of water against the gravitational attraction of the Earth, which you might be able to see in this particular video. But the overall motion is anyway a dissipative positive entropy system because most of that water is flowing downhill, not uphill. So again, it's not contravening the second law of thermodynamics. It's not going against any kind of natural law. It's a net system. It's still net positive. The other concept that we should keep in mind when we're considering living systems is that it's not about the single molecule, it's not about the single object in itself, but the system, the reaction network, to use Pross's definition. So life is a dynamic reaction network which represents different molecules working together, and we shouldn't fall into this trap of thinking about the single molecule as being alive. In fact, when I wrote this paper, one of the comments from the reviewers was that if we can talk about heat engines, differences in temperature doing work, well, that's what my fridge does. So my fridge is alive by your definition. And that's obviously very, very silly. Well, yeah, it is very silly because it's not the single heat engine that's alive, but the network, the different components of the reaction network that are working together to do various jobs, various roles together. So when you think about life and living systems, it really is a system. It's the reaction network, as Pross pointed out. So let's take really the key traits of life. Again, this starts from Pross because my first point here is that life is a self-regulating process. And in this phrase, self-regulating process, there's actually everything that Adi Pross said. In fact, I've put in parentheses that a self-regulating process, this is a self-sustaining, kinetically stable, dynamic reaction network. So everything that Pross said, all of these characteristics of life are compacted into that single phrase, a self-regulating process. But what we're adding here is the fact that the matter that's involved in living processes undergoes cyclic, repetitive, uniplanar conformation state changes. So uniplanar, in one plane. So this could be a revolving enzyme, for example, the ATPase, or a linear system in which the conformation state changes are anyway on one plane. So the matter is changing conformation in one plane. These state changes, the conformation state changes, convert thermodynamic disequilibria into directed motion. So you have differences in agitation states, which are, via these molecules, converted into a directional motion. This motion performs work, and that work then creates the structure. It reduces entropy locally, and this comes back to Schrodinger's considerations of reducing entropy. So with these 5 key concepts then, I want to literally take these 5 key concepts and meld them together to produce one statement, which is the definition of life according to all of these concepts. So here we go. We take all of those phrases and we have a definition of life. Life is a self-regulating process whereby matter undergoes cyclic uniplanar conformation state changes that convert thermodynamic disequilibria into directed motion, performing work that locally reduces entropy. So everything that we've discussed so far is in there, all of these concepts. That also leads us on to another definition, the definition of a living thing. So if I point at an object and I say that is a living thing, it's because it has particular characteristics, and those characteristics we can see here. So this is a definition of a living thing. A living thing is a structure comprising, at least in part, an autonomous network of units exploiting thermodynamic gradients to drive uniplanar conformation state changes that perform work. You'll notice that I put at least in part because not every characteristic of a living organism is actually alive. For example, I have hair that is dead, my fingernails are dead. Not everything about me is actually alive, but there are large parts of me or of a dog or a cat or the tree in which there are autonomous networks of units which are exploiting thermodynamic gradients to drive uniplanar conformation state changes and doing work. This also leads us to a definition of death, or to be more precise, non-life and death, because of course, in order to be dead, something had to be alive before. So as a definition of non-life, a lack of coordinated directed motion in matter reflects a state of non-life. And where directed motion was previously evident in a molecular network, this lack essentially determines death. So we have a definition of life, a definition of a living thing, a definition of non-life, and a definition of death. Now, usually when people present definitions of life, there are exceptions. What about this? What about that? And now what I've done here is to get the usual exceptions and show how to interpret these in the context of this definition of life. And you can see that actually this time it's a lot clearer. Things are definitely alive or definitely not alive. So the first example here is quite easy. Fire is often presented as an exception. It's something that's a self-sustaining reaction. It can reproduce, it can spread, it can grow. But if we consider it in the context of this definition, it's not alive because it increases entropy, it increases disorder. So it doesn't even fulfill the basic requirement of Schrödinger to reduce entropy. So we can state fire is not alive. Okay, fine. Crystals, diamonds, table salt, and snowflakes, these are all crystals that are formed using different processes. They do create structure. They do locally decrease entropy during their formation. But the crystallization doesn't use the same kind of mechanisms that we've seen in cells. It doesn't use autonomous networks of heat engines. These are the results of compaction at high temperature, precipitation from a solution, or by freezing of vapor. So they're very, very different processes, and they don't actually agree with the definition of life. So diamonds, table salt, and snowflakes are not alive. Cyclones are heat engines. There were used differences in thermal agitation between different areas, and they do have a structure, but that structure is created by pressure and temperature differences rather than being a product of the molecules from which they're composed. And they don't form stable autonomous networks in the same way as molecular machines in a cell. So cyclones are not alive. Here it gets interesting. Frozen bacteria or tardigrades, these very small animals, they are alive because they do have a metabolism which is based on all of the effects that we've seen, the reaction networks that we've seen in biology. It's just very, very slow. They're frozen, but those processes do actually proceed. The cell components are in a protected state, which is known as cryptobiosis. This is a well-known biological phenomenon. They are alive because they have that working reaction network. Red blood cells, or erythrocytes, have an active metabolism. They need to maintain the integrity and the functioning of the cell membrane. They do this with a cytoskeleton, exactly as we've seen before. They need to maintain the integrity of the hemoglobin proteins that are transporting oxygen. So they do have a cytoskeleton. They have the ratcheting motor enzymes like we saw. And it's true that in the case of mammals, erythrocytes have no nucleus, so they have no capacity to replicate, to synthesize more proteins. But they are alive in the sense of the definition. We could perhaps see them as senescent. They're alive in the moment, but they are losing function and slowly dying. We could say the same thing about neurons. The neurons in your brain are alive because they're functioning using an active reaction network, which uses all of the principles that we've seen in the definition, but they cannot replicate. Neurons have a certain structure for their job, which means that neurons cannot replicate. It means that as soon as your brain is developed, it can't maintain itself and it gradually dies. This is a process of senescence. But I think that you would agree that neurons in a human brain are alive and that your brain is alive. And actually, if we consider life as an instantaneous process, moment by moment, it does agree with the definition that we've seen. Prions are often touted as an example of a biological molecule that could be alive. Maybe it's alive, maybe it's not. Prions are a very particular case of a protein which is misfolded, so the shape is wrong. And it's true that when that happens, it can induce a cascade of misfolding in other prion protein. But that's not replication, that's not reproduction, that's a cascade of failure. And in fact, prions become structurally and functionally crumpled up and more solid, and they don't actually do anything, like unlike the biological molecules that we've seen. So it's not true replication, and they're not really doing anything. It's just a loss of structure and function. And that actually represents an increase in entropy, which of course is against the definition that we've seen, where entropy should decrease. So prions are definitely not alive by this definition. Viruses are an interesting case because when they're in their free state in the environment, they're simply aggregates of molecules. They're not really in a living state, they're not doing anything. When they encounter cell membranes and they are incorporated into metabolic machinery, they do actively participate in this directed motion network. So they share the living state of the cell. This does reduce entropy by converting simple resources into more complex copies of the virus particles. So it is a living state, but I think that this is the living state of the cell. I think that the viruses in and of themselves are not actually alive. They just plug into the living state of the cell. So if you'd asked me a couple of years ago, are viruses alive? I would have said that it's a complex issue because there is definitely a phylogenetic tree of viruses. Viruses can evolve. They could be considered living entities. But I think here, if we use this definition of life, viruses in and of themselves are not alive, but they can participate in the life of the cell. Okay, another situation in which we might need to know if something is alive or dead is the complex situation of having a dead person with live organs. And this comes back to what I was saying about an organism being a mosaic, being a mixture of living and dead components. You can have a dead human where one of the organs, the brain or the heart, has an intact and working reaction network. Okay, so in that particular sense, in the sense of the definition, the brain or the heart can be alive, but the entire organism can be considered dead. So this does actually fit quite well with, for example, the legal determination of death or life based on the activity in the heart and the brain. And all I'm saying here is that it does actually have a basis in biology. I'll touch very briefly on the subject of artificial life because in the paper I did discuss this quite a bit more, but today I want to concentrate on another aspect of life. Let's just say that this definition that I presented is based on matter and energy, and sometimes that might not seem relevant to artificial life, which could be based on software, for example. But even software has a physical basis in the machine it's running on, and that machine also has an entropy state. So these kind of considerations are relevant. I invite you to look at the paper for a wider discussion of this, but today I want to concentrate on another aspect of life, which is life on other worlds and identifying life. So the idea here is that the definition of life is not based on any single chemical feature. It's not based on DNA. It's not based on ATP. It's based on a physical principle of heat and how atoms move, thermal agitation. And this is a universal thing. And I literally mean universal in the universe. So essentially, the main principle which underpins life is that random thermal motion is being directed by molecules into a directional process. So it's directed motion that can do work. That is a very simple process, a very simple phenomenon, and it's something that we would expect to find anywhere where there is thermal energy, which means basically most places in the universe that are close to stars. So one of the points that I'm making here is that these simple physical principles are independent of any specific chemical environment. It's not something that's specific to Earth, for example, and they could be applied to other biospheres. And we have many candidates where these kind of thermodynamic principles could be very evident. For example, wherever we have liquid water, wherever we have a medium where the molecules are banging around, they're thermally agitated and they can be used to do work, we could potentially have life. These are two examples from our solar system. We have Titan, which is the largest moon of Saturn, on the left, and we have Europa, which is an icy moon of Jupiter, and they both have, as far as we can understand, they both have liquid water oceans underneath their outer shells. So this is a place where there are organic molecules, there's liquid water, there's thermal agitation, and potentially we have molecules moving around. And so long as they're in a configuration where that movement can be directed, there's a good possibility that we could find some kind of living process that agrees with our definition. So because the main requirement for life is a thermal bath and thermodynamic energy, this is something that we would expect to find basically around most stars. Many star systems now are known to have planets in what we call a habitable zone. The habitable zone is usually defined by a temperature which is appropriate for the presence of liquid water. There's an assumption here that liquid water is essential for life, but it could actually be other kinds of liquids, other kinds of thermodynamic media. But let's just stick with this view of habitability as being based on the presence of liquid water. And we've actually detected many star systems in which a number of planets are in this habitable zone. So they're the right distance, knowing the intensity of the star, the distance of the planet, the mass of the planet, we can work out if there's likely to be liquid water or not. And I just draw your attention to the fact that being in that habitable zone is not the only circumstance in which we could have liquid water. We've already seen from our solar system that there are worlds essentially that are outside the habitable zone, which can have a liquid water ocean. So these are examples of star systems compared to the solar system in which there are habitable zones with planets occupying that space. We actually have a lot of data about hundreds of planets that are the right distance, they get the right intensity of light, the right temperature to have liquid water. So there are probably many, many, many thousands of planets which are habitable in the universe. And what I want to do now, just as a fun thing to wrap up this seminar, is to consider what the surface of these habitable alien worlds might look like. And in order to do this, I basically asked an AI, with all of the keywords from this talk, I asked an AI to come up with some pictures of habitable worlds, what an AI thinks that these might look like. I had a choice. I could draw it with a pen and paper in my own non-artistic way, or I could ask for some help from an AI. So what I'm going to show you now is an AI view of what habitable alien worlds might actually look like. Not something with flying cars and big glowing mushrooms, the kind of thing you'd see in Hollywood, but based on simple physical principles, assuming that this definition of life based on thermodynamic principles is a widespread and highly probable principle. So the first view that we have, what did the AI give us? Well, this is a possible surface of a habitable planet. It looks like an ocean on Earth. The words that I suggested to build this image were liquid water, water vapor, clouds, and it came up basically with this. This could literally be the surface of a habitable world. We can see that the sky is blue, so there's probably oxygen in the atmosphere. That's a first clue that in this fantasy AI habitable world, there are probably photosynthetic organisms in the ocean producing oxygen. So that's the kind of thing that we might actually expect if we went to another planet and we saw signs of life, such as oxygen being produced. This is also literally the surface of a world where we might expect to find life. This is not an AI image. This is actually a composite photograph. Now, this looks maybe like the surface of an alien planet, but it's actually Earth. This is the Yellowstone National Park, as painted in 1872 by the artist Thomas Moran. And I've used this as an inspiration for how a habitable world with bacteria, with single-celled organisms, without complex multicellular life, how that might look. So I took keywords involving Yellowstone. I also threw in the word Zabriskie Point from Death Valley, the kind of landscape that I might imagine life to occupy on another planet. And I threw in some keywords to do with bacteria. And let's see what the AI came up with. This is, according to the AI, a realistic representation of an alien planet in which we have single-celled organisms occupying water. There's a lot of water in the atmosphere. We can see that. We can see a storm, and we can see the lightning. This is another AI image, which is a realistic representation of a habitable world in which we have single-celled organisms. So this is not a photograph of Earth. This is a photograph of the Yellowstone National Park. It's a photograph of the Opal Pool. This is a photograph of filamentous bacteria. So this is the kind of structures that we might expect to see if we went to the surface of an alien planet and we were expecting to find life. In fact, for identifying life, if we have a sample from another planetary body, we bring samples back from Europa, for example, or we go to another planetary body, we might expect to see organized structure. Okay, so obeying Schrodinger's principle of reduced entropy, organized structure, a suite of components that operate on the heat engine principle. So all of those thermodynamic principles that we've seen, for example, the motor proteins, how they work. And this is really important. This is the bit that usually gets forgotten. All of that has to be happening in a thermally agitated medium. So in water, for example, or some kind of liquid where the molecules are banging around, and that energy or differences in the state of that energy can be used as an energy source. So what I did here was I generated a set of AI images based again on the keywords in this talk, such as cytoskeleton, thermodynamics, even pollen, which was a big part of discovering Brownian motion. I put all of these keywords in and asked the AI to imagine the kind of life that we might find based on this definition. So I have some images that look kind of like cells or the kind of structures that we would see under a microscope, but this is all kind of AI fantasy, okay? I'd like to underline that. These are not real structures. But if we did go to another planet and we saw some kind of structure and the components were moving, they were working together, and they're in a thermally agitated medium, this is the kind of thing we might expect to see. And seeing that working is really the only way to be sure that we've seen life, that we found life. So especially if we can get this kind of image, obviously I've manipulated an image from an Earth organism, but if we can see things moving and working together, that's a really strong indicator of life. So recently there's been a lot of press about the fact that the James Webb Space Telescope has basically discovered a molecule called dimethyl sulfide, which is only produced on Earth by microorganisms in the sea. And this is a strong indication that there could be life on this exoplanet K2-18b. So in the press, there's a lot of talk about the fact that we could actually have discovered life on another planet. I invite you to read the original paper where they discuss the importance of this observation. Okay, cool. We have an indicator that there could be some living process on that planet. but we need to see the actual thing itself, because we need to see the components working together, reducing entropy. We need to look at all of the processes that happen in the definition of life. For example, if the Perseverance mission returns samples from Mars, what we might expect there is fossils or some kind of chemical system, but we won't necessarily see it actually working. And I think these would be strong indicators of life or past life. But in order to be sure that we have identified life, we must see the components working. We must see the molecules moving, and ideally they should be in a context which is natural, which is their natural situation. So a thermally agitated medium. Only when we've seen a complex chemical system where a reaction network is working together to reduce entropy with this characteristic that the molecules change shape in a directional way, only when we see that kind of situation can we be sure that we've really identified life from another planetary body. I'd just like to leave you with one last thought, which is a very, very speculative thought. I don't want you to read too much into this, but that's about the existence of civilizations in the universe, because basically what I'm saying is that life, simple life, is probably very, very common. It's based on a simple thermodynamic principle, which seems to be a characteristic of hundreds and thousands of planets that we are detecting at the moment. So simple life is probably extremely common in the universe. Civilizations, this is a completely different question, but I'd just like to point out there is an example of a civilization in the universe, and that's our civilization. This is important. It's not as dumb as it sounds, this, because it means that we have a precedent for expecting a civilization in the universe. But watch out. Earth is over 4.5 billion years old. For most of that time, 4 billion years, probably around 4 billion years, there have been at least single-celled, simple life forms on this planet. So for the vast majority of the life of this planet, it has had life. It's a planet that occupies the habitable zone of its star. But only for the last 80,000 years or so, based on artifactual evidence, has there been some kind of technological civilization on Earth. And by technological civilization, actually, I'm just referring to the fact that we've found a few small artifacts from around about 80,000 years ago. So anyway, this is a very, very small period of time with respect to the whole period where Earth has been occupied by life. So what that means is that even if we find 50,000 planets in their habitable zones, it's very, very likely that they have some kind of simple life, but extremely improbable that they have civilizations, because even on Earth, where we do have a civilization, that only really came about through a series of extremely improbable events very recently. So even Earth basically has been characterized by microbial life for much of its existence. So this seminar is based on a peer-reviewed paper, which I published last year, Life's Mechanism, which you can download from this web address. I'm going to leave the summary, the abstract, on the screen for a while so that you can read it. But if you want to know anything more, if you want any more details about the very particular processes that we've seen during this talk, you can refer to this paper. Thank you very much for watching. Goodbye. Grazie.